Hi there, so I'm Catriona Crombie and I'm Head of Rare Disease Research at the medical research charity LifeArc. Now we're a charity that works very much just in the translation space, and by that we mean we work with basic scientists to try and get their science out of their basic research lab, do whatever is needed to that research in order to take some risk out of it and hopefully get somebody else to take it on and take it all the way to market and deliver some real impact for patients with that innovative science. So that's what we mean by translation. But I think this project brings another element of translation together. When my marketing director came to me and he pitched this idea about getting scientists and artists together, and I thought, gosh, how is this going to work? How are we going to turn some of this really detailed science into pieces of art? And that's what these guys are going to tell you all about today. And it's been a fascinating project. But it's a different sort of translation. It's taking that language of science and art and bringing them together and actually having that dialogue and collaborative working to bring that together. So I'm going to quickly hand over to the guys that are really important. We're going to start with Gavin and Charlotte, and they're going to tell you about their partnership and how they made a phenomenal piece of art.
Yeah. Right, so I'm going to start off by just saying a few things about the overall project and then hand over to Gavin for a more in-depth discussion about the science aspects. So art and science collaborations, and I'm sure you'll very soon realize, take many different forms. And this particular project that we have been doing has actually evolved quite significantly over a period of time. So it started back in February when we were matched together by LifeArc and CamRARE. And at that point, in February, Gavin told me about his research into the functioning of the mitochondria. I then, as an artist, began to explore both form and function through ceramics. And in doing so, it became apparent that the functioning and behavior of the mitochondria is a source of both strength and vulnerability for all of us throughout our lives. So fast forward to I think, spring, we began to focus much more on the vulnerability aspect of the mitochondria. Having focused previously on the functioning, we began to address the dysfunctioning mitochondria and what that means to us in our daily lives. So at that point, we were really fortunate to be able to, what are they called? What are they called? The Lilly Foundation. Sorry, I just lost the, to be, connecting up with the Lilly Foundation, who is a foundation that supports patients with mitochondrial diseases, but they are also very keen on making sure that we all know more about the research into the mitochondria and how it affects our lives. So fast forward again, and we're in the summertime, and Gavin and I presented our project on the art science project on the mitochondria to a patient group from the Lilly Foundation during their first ever adult support weekend, where we then met and spoke with patients and medical practitioners who look after patients with very rare mitochondrial diseases. Those stories of both patients and medical practitioners have now found their way back into my work, the artwork, where they sit alongside the science part of the project. And I think it's really important both to have the lived experiences as well as the experiences that you come across in the lab and observe. And we have had a lot of dialogue about the commonalities of those two aspects. So I'll now hand you over to Gavin for a more in-depth science, for the science aspect of this project. And at the same time, I'm going to hand out a couple of little porcelain structures for you to pass on amongst yourselves, so you can experience the sense of strength and vulnerability that is inherent to the mitochondria.
Okay.
So that's it. Over to you.
Okay, thank you, Charlotte. So Charlotte refers to me as her scientist. I am, but it's because we've had a really good relationship since we've been matched. So it's this work has really given me something to think, gives me an extra dimension to think about my research on mitochondria. So on the screen here, you can see some of what the mitochondria look like that Charlotte has made. So they are kind of handheld items made from porcelain that have different colours and different shapes. So what I'm going to do is explain what we're trying to represent with these structures and the colours within these mitochondria. So the mitochondria, we've called the work, or Charlotte called the work, the flame of life, meaning the mitochondria are providing us with energy, giving us almost like they provide a certain amount of heat to us, but they provide function and energy. So this is where the title of the work came from. The mitochondria, as probably many of you have heard at some point in your life, are the powerhouse of the cell, meaning they provide energy, they provide us with the ability to do the things we do every day in our lives. So every cell in our body at some point has had a mitochondria in it, or has a mitochondria in it that is fueling its activities, and that's fueling us to be able to do what we do day to day. This has been a very traditional view of mitochondria. So mitochondria are very popularly researched in the mid-1900s as an ATP energy-producing part of the cell. But over the past 20, 30 years, the functions of mitochondria have become much, much broader, and now they seem to be involved in all kinds of things related to immunology, neuronal function. Most of biology is fueled by energy, and the living parts of it are, a lot of mitochondria have got something to do with it. So what's really interesting about mitochondria is their origin. They are derived from a bacterial invader of a single cell back in evolutionary time, and this has given rise to a very distinctive structure, which is what you will be able to see on the screen, but also hold in your hand. So the two images on the bottom represent how mitochondria have been represented over the course of time. So on the bottom left, we have a very, very classical image of what mitochondria look like. They are a bean shape with lots of little kind of crossovers in the middle, which represent the functions of those where the mitochondrial function happens. And then next to it, on the right of it, in this purple, blue, and yellow image, is how we look at mitochondria these days. They are a network. They're very, very active. They're very dynamic, and they're very mobile within a cell. So they're doing lots of things, and they're very, very busy. So a mitochondria itself is a very, very complicated thing to make, as in most things in biology, but a mitochondria has several distinctive features that make it more tricky to make than other things. It's got two membranes, so in the mitochondria that you're holding, you have the outer membrane and then the inner membrane, which represent the two distinct parts of the structural parts of the mitochondria. And then within it, we've got different areas where different biological fluids exist. So within the mitochondria itself, it's made up of many, many different parts, many different pieces, and involves many different steps to make it. So before you make a mitochondria, you need to make sure you've got everything that you need to make it. So if you look slightly left to my first point there, that is the instructions that you might, or like the inventory that you might get if you were to build your own furniture, if you were to buy it from a major seller of wooden furniture. I don't want to say the name. So you want to make sure that you've got everything you need to make it. So the cell needs to check that it's got all the parts to make its mitochondria before it even starts making a mitochondria. Otherwise, it's just going to waste its energy. To make this happen, there are many helpers within the cell and many signs that direct in which way the mitochondria are being made. So there are lots of proteins and lots of genes that are responsible for assembling really important parts of the mitochondria that make energy, and there's lots of signs. So you have to make sure that the helpers are working and the signs are pointing in the right way. Otherwise, again, the message and the instructions are not clear. What makes mitochondria even more interesting, and everything in biology, is three-dimensional as well, and it's got different places, different locations. So again, you have to send things to the right place at the right time, in the right directions. And it has to be done in the right order. You can't, if you imagine building your wooden furniture, if you start from one end and you think, oh, I know it's better to do it this way, actually it doesn't, and you have to then dismantle it and then rearrange it again. So it has to be done in a very specific order. So for me, the way a mitochondria is made can be, you can think of it as like either being a journey. So I came here on the train yesterday a different way. I'm going home a different way tonight, and it may be impacted by some bad weather. So that bad weather could reflect things that could be a problem as a mitochondria is being made. The journey might have to go a different way. I might need to ask people, how am I going to get home? So within the mitochondria, there are these checkpoints to make sure that it's happening. It can also be seen as a puzzle or the flat pack furniture analogy that I mentioned before. So if we think of it as a jigsaw, if you think about how you see a jigsaw and how you make a jigsaw, we're never given any instructions really how to make a jigsaw. We just see what the final image looks like, and then we get a pile of pieces. I don't think we can ever really, if there's 1,000 pieces of that jigsaw, are we going to count that we've got 1,000 pieces at the beginning? I don't think so. Maybe if it's a simpler jigsaw, we can do that. But in instances, there may be cases where the jigsaw is missing pieces. So maybe if you buy a second-hand jigsaw, you may not have all the pieces. You may be wrong pieces within that jigsaw. There may be too many pieces in the jigsaw. There may be not enough helpers making the jigsaw. So for me alone to make a jigsaw would take a longer time. If Charlotta and we do it together, it happens faster. So it's a more efficient process if you've got the right number of helpers. Sometimes if you have too many helpers though, too many chefs in the kitchen, too many little people who want to be involved but maybe not helping in the right way, this can influence how fast it's made as well. And the helpers are not helping. So it's there has to be a very specific way that this is controlled and managed in the cell. So how this relates to diseases and rare diseases specifically associated with mitochondria is that if there are changes in gene expression, so if one piece or one of the helpers is not functioning properly, is absent, this will influence how the mitochondria is made. And this could be because there are mutations in the genes encoding for the mitochondrial structure itself or the helpers, or the cells are generally not healthy, so not even caused by a mitochondrial mutation, but just in a general chronic disease, this can also be impacted. And this will then lead to your mitochondria being made incorrectly. So if our mitochondria are made incorrectly, they don't work properly, the cells and tissues don't work as they should, this can then lead to disease. These diseases are very difficult to diagnose and treat, so this is one of the main issues with mitochondrial diseases. Another really important aspect of mitochondrial disease is the way that they're inherited. I won't go into any details about this, but it's not a both parent contribution, it's only through the mother, so this makes it very difficult to kind of track how it's going to be inherited. And there really needs to be an increase in awareness of these diseases because mitochondria contribute to so much in our day-to-day lives. So the mitochondria that you see are representing different states of mitochondria here. So on the left, we've got healthy mitochondria, in the middle, low functioning ones, and then damaged ones, and then mitochondria as their network. So in mitochondrial diseases, they are often poorly functioning or damaged. So these are represented by the low-coloured mitochondria. And so this is where the flame aspect of their function comes from. Okay, and so just want to kind of highlight these, kind of how our art and science collaboration has made me think a little bit differently about mitochondria, is that the mitochondria represent differences in strength and vulnerability in the mitochondria, so how Charlotte was making them represents things in the process of making a mitochondria in the biological scenario. So this really we felt a synergy there, that there was, what Charlotte was doing was something that actually the cell is trying to do as well. It's a process that needs to be done correctly. And then also mitochondrial diseases are an individual disease. Every mitochondria can have slightly different variations in its function because of genetics or the situation. And so the mitochondria that Charlotte has been making are individual as well. No two really came out the same. And so mitochondria and mitochondrial disease patients also feel like they are unique patients as well. So that's where we would like to finish, and we'll pass on to. Thank you. Thank you. Thank you to Kate and Donna.
Brilliant, thanks very much.
Straight on.
Kate and Donna are going to talk, and then we'll come to the audience after Kate and Donna have done their little discussion about how they came up with the art, and then we'll come to you for some questions. So have a think if there's anything that you want to ask, please.
Thanks. I'm Kate. This is Donna. We'll introduce ourselves in a second, but it's just been so exciting to create something together this year in a new way. That I'd not done before, and I don't think that Donna had done before. And today to come and talk about what we've made together is exciting, but it's quite nerve-wracking because I know that there are other artists and scientists and families and patients here as well. So talking about what we've made is quite daunting. So let's introduce ourselves. Donna, would you like to go first?
Yep, I bring with me to this project my naturally enhanced tendency to see faces and perceive faces and creatures hidden in inanimate objects and unusual places, which is called pareidolia. And so you can see on the left is a glass of kombucha, a cup of kombucha that I was drinking, and I drew what I perceived in the swirls. And on the right-hand side, it is my drawing, which is inspired by a photograph taken by Martin Bond, and I just perceived so many different things in there, faces and creatures, that I wanted to try to share how I see the world. I mean, for example, there's a very big spider in there, and I'm terrified of spiders, but you know, my son said, well, that's the size of a bird-eating spider. And I said, well, he said, you're gonna have to draw it. It's in there, you're gonna draw it. And I do try to be true to what I perceive, so that's what pareidolia is. I bring that to the project.
So as you can hear, Donna's an amazing visual artist who's able to translate what she perceives internally, how she sees the world, which is different from anybody else's, into something that communicates her perceptions to the rest of us in a very beautiful way. What I do is I'm a clinical geneticist. I'm a doctor. I work at Adam Brooks Hospital. And some of you may not know what clinical geneticists do. We sound quite technical. It sounds like a very technical part of medicine. But actually, clinical genetics is a medical specialty that's really all about communication and translation. Because what we try and do is we bring together the questions and the problems that a patient has and that their family may have about their health or their development. We bring together that information and those questions from patients with information about their genome and match that together to see if we can make a diagnosis of a rare condition. And making a diagnosis isn't just something that comes out from a sequencing machine. It's not just a bit of code. It's actually a communication exercise between what has a patient experienced, what do we see as doctors and as practitioners, and what's the information within the genome, and how can we make sense of that together. So it requires a lot of communication. And the other parts of a diagnosis is even once we've got the name of a condition, a diagnosis is only useful if it provides a new way of seeing somebody's experience. So a new way of seeing what's happened in the past, a new way of understanding what's going on now, and some new ideas of what might happen in the future. That's what a diagnosis really is. And again, that involves some science, but it's really about communicating and connecting and understanding what's important to each patient and their family going forward. So that's clinical genetics. And I find this process really fascinating, but also really frustrating, because a lot of times patients will ask me questions that I don't have answers to. And the role of all medical practitioners is to get the best possible answers together with patients. And that's why a lot of us do research as well. So I run a research group here at the University of Cambridge, and we're actually in the neuroscience department, not in genetics. We want to understand what happens after diagnosis. We want to understand how is it the case that these tiny little variations in DNA sequence can affect how brains develop and change over life and alter things like perception and control of speech and control of action and emotion regulation and ultimately relationships and mental health. So my research is always all about trying to understand people who do have a genetic difference causing a rare condition and understand how that manifests in changes in brain development. And that's some of the work here. We can do, so the very top left is a cartoon of one particular gene called synaptotagmin 1. And that's one that I've worked on for 10 years now as a researcher, because I was part of the team that first discovered that differences in that gene could cause a rare condition. So we discovered a new disorder, which is an interesting thing to be part of. But the story doesn't stop there, because you can map the gene differences, but what we want to understand is how can that difference alter the dynamic, complex, cascading processes of brain development to influence perception and motor control and emotion regulation. And that's why we've still got a lot of work to do after discovering a new genetic condition. So we were matched together. Donna, with her amazing drawings about perception and about faces, and my work as a clinical geneticist making diagnoses and then trying to get better answers for patients after diagnosis about how these gene differences are actually causing the symptoms and the experiences of patients. So how are we going to make a piece of art together? Donna had an idea. Do you want to explain the idea, Donna?
So this is the first sketch that I did for the drawing, and I think Kate looked at it and thought, this isn't going to go well. But actually, I'd just been to see the organ scholar at St. John's College, Alex Robson, and he'd spent a couple of hours time with me while he was rehearsing, and it was just awe-inspiring. And it's pretty much how the drawing looks, the way it's laid out, amazingly. He's there in the front, and then on the right-hand side is the backstage, backstage of the organ, which is a lot of infrastructure, surprising amount of infrastructure, and almost completely mechanical as well. So we saw a correlation between that kind of complex structure and the structures of the human body, and that was really something that brought us together. And on the left-hand side is on the left-hand side is the chapel, where you would come to see the front of house of the chapel, and I had put in the organ the choir and the director of music. And also at the top, when I was there, I had such a wonderful experience lying on the floor, looking at the ceiling to try to figure out how to show this building, this amazing space well, while Alex was playing the organ, and I had my eyes closed. And when I heard the sort of twinkling sound of the music, I opened my eyes, and he had this star, this gold star, the Zimbalstone spinning, which was just magic. So, yeah.
So as you can hear, Donna and I, when we started our collaboration, were actually thinking about very different things. I was thinking about genes and brain development and mental health and families. Donna was thinking about St. John's College Chapel and sound in the organ. Whoa, how were we going to bring those together? So we had to talk a lot and listen to each other and learn. And initially, I couldn't really see that connection. What was the bridge going to be? But in our first meeting at my research department, we shared our ideas, just as we've shared them with you today, and started to think creatively, move our ideas forward by listening to each other and see things and think about things in a new way. So we made sort of two decisions, really, at our first meeting about how we could bring those together. One was to think about how Donna's art relies upon her special abilities of seeing faces in structures. And I explained how one of the things we do in clinical genetics when we are making diagnoses is match together the physical characteristics of patients, including their faces, with known syndromes or information about someone's genetics. So there was a link there, actually, about the importance of faces in rare disease diagnoses. That was one decision that we made, that had to come into the picture. And the other decision was that we would involve members of the CamRARE unique community in this piece of art, because this couldn't just be an artist and a scientist trying to build bridges. It had to be something about actually involving rare conditions patients for this to make sense, to have a purpose. The other decision was that I still didn't understand what Donna wanted to make that was to do with organs and the chapel. But Donna convinced me that in order to understand what she wanted to create, I needed to go with her to the chapel to Evensong. And I went with my students and had to experience, let go of a lot of these words and ideas, and just experience the space, just experience the sound, just experience the light. And I think as scientists and doctors, we expect that words will have all the answers. And sometimes they don't. We have to just let the feelings take over. So that was a special experience.
Yeah.
And it allowed us to come to a critical kind of insight that I'd not had before, I think, or an idea, a thought, which was again about the organ. So the organ in the chapel was critical to what Donna wanted to draw. And I realised that one could link the organ to thinking about the human genome. So there are 20,000 genes in the genome, and variants in many thousands of those genes can cause rare conditions. And when we try and find a diagnosis, we focus in on just one tiny sequence change that might be the cause of somebody's rare condition. And I realised that we really needed to think about the genome like the whole organ. And if that there's one gene variation somewhere in the genome, a bit like a slight tuning difference in part of the organ, that difference in the organ or the genome is not acting on its own. It's part of this big interactive structure that comes together to generate life and health and a person's experience. And we need to sort of zoom out and understand how those gene variants are part of this much more complex and beautiful system in understanding what a diagnosis really means. So that was a thought I wouldn't have had if I wasn't in the chapel with Donna.
And it was interesting that there's that so little of the organ. Can you go back just a second?
Yeah.
So little of the organ that you see in the chapel, and we know that there's so much behind that. So it's that's fascinating too.
Mm-hmm. Exactly that. That what we see is only a tiny glimmer of the system.
Now the
underneath.
organist is behind the screen, and there's just one rank of pipes, and there's the Zimbelstern, but there's one. That's just about all you see, and there's just so much more, as you can see in the drawing.
So by this process of talking and going together to experience the music of the organ in this beautiful space, we had a joint idea now about what this drawing was for and what it was about. And then Donna went away to create the beautiful drawing. It's very intricate, and you'll have to come and see it in the exhibition to really appreciate it. But I think what's amazing about it is it creates the volume of the space inside the chapel within which the sound of the organ is experienced by anybody within that space. Yeah. And that's captured visually, the complex experience is captured visually within the drawing.
So this,
Go on, Donna.
This is where I was at the point of the deadline for the brochure. And I don't know that the organisers realised it wasn't finished, but it wasn't finished, and Kate knew, because I had promised I'd also put her and her research team in, and they weren't there. And there's no director of music and there's no choir. And I said, well, Kate, don't worry, I will get finished, you know. So that's where we were at the point of the brochure deadline. And there's no sound, but I always knew I would put the sound in, yeah.
And the later phases of the drawing, the final drawing, is a really amazing embodiment of, I think, this experience of creating something together, because Donna very kindly brought me and my research group into her picture and into the chapel. We're at the bottom there, where we're looking and listening and experiencing the world of rare conditions and trying to understand something more about the relationships between genes and biology and sound and experience. And so it's a really special thing to actually be drawn into that space. Another thing that's very special for me is that within the space, thanks to unique feat participants, there are members of the Cambridge Rare community who are within the drawing. And that's important because they're part of Cambridge, and Cambridge system and everything that Cambridge is relies upon the whole community. But also that there are some of those individuals who I've met as a doctor, and they have had great impact on me in my thinking and in my career. So to share a piece of art together is something really, really special for me. And I'm very grateful to Donna to give us that chance to feature together.
There's a lot of links that we didn't really understand at the beginning, that, for example, St. John's College Chapel did fundraise for Unique Feet not too long before I started the drawing, and then it's just a really nice kind of way to say thanks again to them for giving. They raised funds for some money for Unique Feet to do some activities, and also I had gotten involved with a sound acoustic engineer. He's an architectural acoustic expert called Dr. McLuskie, and he's local, and he went to the chapel with me, and we listened to an organ recital, and he was telling me how he heard those red notes of music coming over, and it was a lovely experience. And he actually was involved with St. John's years ago when he took the choir to Italy to sing music of the period in the churches that were built at that time, to see how the acoustics were back then. So it's really interesting how you have all these people you're collaborating with and you think this is too complex, and actually it just adds layers that are so beautiful. I would not hesitate to collaborate again.
So altogether, this piece of work is called RAREfest. So it's about what we share in Cambridge as people who live here, as citizens of the city, as artists, people who've had medical care at Adam Brooks Hospital and the university, and these special spaces like St John's College that we can all come together and share. And that's also something that's been captured. So thank you so much to Donna for working together, to Alex, who's the organist in the piece, to Raf, who's the sound engineer who helped to put sound into the visual of the picture, of course to CamRARE and Unique Feet and LifeArc, and my research group who supported us and are in the picture too. And I hope that more people within Cambridge and beyond can have the opportunity to connect and communicate and create pieces of art together, because it's a very special experience and it doesn't come along too often. But I'm very grateful to have had this chance this year and hope more people do again in future. Thank you very much.
Yes.
Thanks, Donna.
Brilliant, that's fantastic. It's really interesting to hear about the process that you went through to come up with these amazing, unique pieces of art. I'm going to come to the audience in a second, but I'm just going to start with a quick question, if I may take chair's prerogative. And just ask you, in terms of the partnership that you had, has it changed the way you think about your research going forward? And maybe I'll ask that to Gavin first, and then I'll come to you, Kate, and think we can...
Yeah, so Charlotte and our conversations we were always asking questions of each other. So Charlotte is asking how do mitochondria work? How are they made? So it made me go back to the biology and think about how these processes happen. And so some of these questions sparked little questions in my mind that I hadn't thought about for maybe since I learnt about the processes of mitochondria 20 or so years ago. And so it kind of brings things back into my orbit in terms of how a mitochondria should behave with kind of physical forces around it as well. So the manufacture of a mitochondria physically in 3 dimensions at a macro scale, how does that relate to it in a the micro scale within a cell?
I think for me that was because some of the early questions I had was what does it look like, and of course it's the shapeshifter that actually looks like all sorts of different things at different times depending what's going on. But we decided to stick with the bean shape with the 2 membranes that people are used to seeing historically. But my questions to Gavin quite early on was what is the strengths and weaknesses of this bean shape? Because I can see in ceramics it breaks really easily when it's like this and during these stages. So that's a sort of a physical element of a shape that you were sort of coming back to. Oh, I'll have to have a little think about that and get back to you, and we'll need to see what we can, yeah.
Yeah. So
if we have any answers to that one yet.
Yeah. Still in progress.
Yeah, exactly. So, yeah.
Great. I've talked a little bit about how thinking about the organ and Donna's ability to capture the beauty of a whole system has helped to prioritise the research questions that I want to take forward a little bit in terms of sharpening my focus on seeing gene differences in a bigger picture. But also it's changed a little bit some activities that I want to do in helping to prioritise my research questions. Like after we went to St John's College together, I actually did some creative writing that when I came back from the college, from the chapel, it was like a need to write a bit about, not in a scientific way, but reflecting on some of the experiences I have with patients and on the mutual impact of giving a diagnosis and pointing out differences and how that changes perspectives on a person. So anyway, it made me want to write in a different way. And I think that, again, that process, being part of a creative process, helps to take steps forward with research. By doing something differently actually helps you to focus on the questions that really matter.
Hi, thank you so much for being here today. It's amazing to hear all your perspectives on the artworks that you've created and the process behind it. I'm quite interested in the practicalities of the project, really, in terms of like how you found different people to participate. In terms of like, how did you contact everybody involved, and how did you link people together? Because I think it's an amazing opportunity that you've created, and I'm just intrigued to hear about how it happened, really.
So this was an idea that my marketing director came up with. He had worked in a previous job with Joe on a very similar project, and he got Joe involved and Gary here. And so I'm going to throw this question at Gary, because he's actually the man who pulled all of this together, and I definitely can't take any credit for it. So could you just give us a couple of words on how you matchmake people?
So we split it up. So Joe was in charge of bringing the scientists and the families together. My responsibility was to make sure we had enough artists, and the questions for me at the beginning of this, because I certainly hadn't done it before and we hadn't done it quite like this before, was just like, are we actually going to have enough people? Are we going to get work that's significant enough before even thinking about the pairing of the two? So all the art networks that I was aware of, we put out an open call for artists, or artists that I knew. I'd say, we're doing this. If you would like to apply, please do. While at the same time, Joe was doing something very similar for scientists in the network, the Cambridge Rare Disease Network as well. So in effect, the scientists, I think, pretty much were an exact match for the amount of people that we needed. I think it was like, oh, okay, we have 20 scientists. The artists, it was a little bit more complex. We had more applicants than we had spaces, so we had to do some kind of like selecting of them, and they had to explain why they wanted to do it and what they were bringing to it as well. What I thought was really important was that we had diverse styles and a diversity in the artists, and on many counts we managed that. There is only one gentleman in the artist group, and that's not me. Hi, Andy, over there. Well done, Andy. But broadly, I think we did have diverse work, and the art itself, I thought, beautifully responded. But the artists had, I think, roughly you can correct me if I'm wrong, because I think that these deadlines impacted you guys more than they did me. 5 weeks to make connections and make the work, is that right? 5 weeks? Something like that. So in effect, we suggested make contact, go talk, and then come back. And it was up to every single artist and scientist pairing to then go about that in whatever way worked for them. And I think lots of people had really different experiences of that. But then once that was in, it was just like, the artist then went and then created, however they'd collaborated, but then went and made by themselves, so that we had the first show in Islington back in April, and then the second show here. Does that answer your question? Yeah, thank you very much. And I thought everyone did great, so I'll just say it publicly here. Well done.
I'm going to ask the audience a question now, because I think that's very important. Are any of the young people that participated in Donna and Kate's piece of art here? And I wonder if any of you would like to say anything about what it means to you to have been involved in this art project.
Everyone would like to say thank you for letting us be in the art, and it does mean that it shows that everyone is on there, whoever's got their disabilities that we have, that it's a good way for people to know that we got disabilities, and we I just think it's amazing that some people in our group that are in the picture.
Yeah, we definitely do. Thanks.
And to meet you.
Oh, well, that's same here.
another question just behind you.
We heard from the medics how it's influenced their research going forward. I wonder how it's affected the artists in their work ongoing.
Well, yeah, so I have collaborated in the past, but really just with like one person. But this was a really big collaboration. I had this determination to show sound, so I started with that personally as an artist, and I decided I was going to find a way to do that. And that's where I began. That's why I think Kate and I had such a disparity at the beginning. She was like, well, I don't you know, but then I got the sound expert in, and that was just an awesome experience for me. And also Kate, working with Kate, she's just, I mean, you can see she's just full of insights and information. And then the organ scholar, I never expected to be blown away. I was like walking around for 3 days in a sort of daze. I just never expected to understand so well the organ and be able to be so inspired by it. So it has really, I'm just moving on. You know, I did 8 colors in this drawing instead of just 2, and I used watercolor ink. And now I did 28 in the next one. And I don't know where it's going to go, but it has definitely progressed my practice.
Yeah.
Thank you.
I think I've done a number of art and science collaborations over the past 10 years, and they're all different depending on what is brought to the table, really. And I think that's a very exciting aspect of it for me, really. So, you know, this has panned out completely differently to some of the other ones. And it's been full of dialogue back and forth, which I really appreciate, and I would very much like to take that through to the next art and science project so that it's an ongoing dialogue between two people from 2 different fields, because I think it enriches both our fields quite significantly when we allow that to happen. So I'd like to thank Gavin for listening to my stupid questions along the way and continuing those dialogues, really. So yeah, so I think there's very many different ways of approaching it, but it's that's the excitement of it.
Yeah, the exciting bit and the risky bit of all collaborations and all art and science, the similarity is you have to go off into the unknown.
Exactly.
And form a connection and go into it with integrity and openness, and then change and develop along the way, and that's really exhilarating. Actually, that happens in medicine, it happens in science as well, and I think having an opportunity to experience that, taking those risks and see what happens is a really positive, reinforcing experience, really confidence building.
I think from an artist's point of view, you're asking about the sort of artist sort of aspect of that. It has allowed me to push the material of porcelain, for example, in ways that I wouldn't have thought of pushing it before. Yeah. And that will inform future practice of, I can actually really stretch this a little bit further, even though, you know, it says in the books of how to work the porcelain, you shouldn't be doing this, that and the other. I'm just going to try and give it a go anyway.
And that's inspiring to us as scientists, that actually we can take bigger risks and try and tackle the most challenging, important, interesting questions, even if we might fail, when we don't know where we're going. And I think that that's something that scientists can learn more from the creative industries and from artists, to really be fueled by your confidence and your ambition and kind of vision about what you can change in the world. That's actually a very powerful message for the science community as well.
I think I would really just like to say thank you very much to everybody who was involved in the project, to all of the artists that are, some of whom are present here today, and the scientists who took part. I think it has been a really exciting project, and you can go and look at the art and see just how interesting and different all the pieces are. It's amazing how the amount of the diversity and the creativity that has come about from that. So, and I'd also like to thank Gary for helping us to organise it, and Joe Balfour, who was instrumental in pushing all of this stuff forward. The other thing I want to say is that there are calendars that you can go and buy, so you can take some of the art home with you, or you can give it as a gift. I've actually bought a couple of them because, as a scientist, I know that my family don't really understand what it is I do. It's all a bit abstract to them. And actually, the art, they can kind of understand that a bit better. So I'm going to use that as a kind of tool to take them, to give to give them as Christmas gifts. It's a perfect time of year to be getting your Christmas gifts in. So please go and visit the calendar stall and take some of this wonderful art home with you. So thank you.
And I'll just collect your bits that I was.
Yes. And can we have the bits of porcelain?
If you have a bit of mitochondria, please pass it forward.
Thanks, Marshall.
Yes. So thank you very much, everybody.